Let's discuss C4 pathway or C4 cycle. That is also a way by which the carbon dioxide is reduced to carbohydrate. So it was worked out by two Australian scientists, that is Hatch and Slack. So who are the, this is also called as HSK pathway, because the scientists are Hatch, Slack and Korschak. Hatch, Slack and Korschak. And here C4 plants are special because they produce, they have a leaf anatomy called cranch anatomy. So that's a cranch anatomy. Now what is this cranch anatomy? That means two types of chloroplast occur. One chloroplast occur in your mesophyll cell and one chloroplast occur in bundle shaped cell. Okay, and one chloroplast it has the enzyme called PEP carboxylase. And this is having Rubisco. Now Rubisco undergoes photorespiration, but if the Rubisco is present in the bundle shaped cell, then it cannot come in contact with the outside environment, so it cannot be disturbed. So what are the three conditions that force the Rubisco to have the photorespiration? Like the high oxygen content, low CO2 and the temperature is high. So these things, if the Rubisco is present in the bundle shaped cell, which is near the midrib portion, the xylem and phloem, and that it cannot be disturbed by the outside fluctuations. But PEP carboxylase is there in the mesophyll cell, which is not disturbed by or not show any photorespiration kind of thing. So C4 pathway, where cranch anatomy is special, and it refers to the presence of two types of chloroplast, as I said. So chloroplast dimorphism is seen, and the active participant in light reaction, that is ATP and NADPH, are here also used up. So step 1 is in C4 plants. The initial fixation of CO2 occurs in mesophyll cell, and the primary acceptor of CO2 is phosphoenol pyruvate. So what is this phosphoenol pyruvate? That is the final acceptor, and it combines with CO2 in presence of an enzyme called PEP carboxylase, and that forms the first C4 compound, that is oxaloacetic acid. So this C3 acid is actually oxaloacetic acid. This is oxaloacetic acid, which is the C4 acid. Now C4 is the first 4-carbon compound is formed. Now this compound OA is transported to bundle sheath cell, where they are broken down and releasing CO2 and 3-carbon molecule. So again they break down and form a CO2 molecule. This CO2 is trapped by Rubisco enzyme, and they carry out the C3 cycle over here and produce the C3 acid. This C3 acid is again transported back to the mesophyll cells, and then again pyruvate it forms. So this way there is a transport going on. The C3 acid, then C4 acid, then this comes to this bundle sheath cell, releasing the CO2, and then CO2 is again used up in the C3 cycle, producing. So both C3 and C4 cycle occurs in this cranch anatomy and in the C4 plants. And C4 plants are highly efficient, very efficient in producing starch. Then they can resist the temperature. So photorespiration is not happening in C4 cycle, as because the position of Rubisco is in bundle shape. But photorespiration is there in the C3 plants, where Rubisco is immediately present in the mesophyll cell and then can be affected by the fluctuations in environment. So there are two kinds of plants occurring in C3 as well as C4 plants. Two kinds of plants are occurring. So C3 plants, you can go through the differences between the C3 and C4. But C3, Rubisco is a major enzyme. But in C4, both the enzymes like PEP carboxylase is also present and the Rubisco is also present. But C3 is not showing any cranch anatomy, but the C4 is showing the cranch anatomy, which is a plus point for this. And C4 plants are very efficient than the C3 in production of starch. Thank you. Go through NCERT, join the offline class.